Ever wondered what PGD is and how it's used to choose the gender of your baby? Welcome to our dive into the captivating world of Preimplantation Genetic Diagnostics, or as it's commonly known, PGD. It's an incredible scientific advancement that offers expecting parents the choice of selecting their baby's gender before conception. PGD is a process that involves creating embryos through in vitro fertilization, then screening them for genetic disorders and gender. It's an incredible testament to how far we've come in understanding the human genome and the power of genetic manipulation. This is a topic that's not just for scientists or medical professionals, but for anyone interested in the wonders of modern medicine and genetic science. We'll be exploring two main techniques used for genetic assessment: polymerase chain reaction, also known as PCR, and fluorescent in situ hybridization, or FISH. So, are you ready to delve into the world of genetics and the choices it can provide? Stay tuned to understand the science behind this fascinating process. To fully grasp the concept of PGD, we need to delve into the basics of genetics. Let's imagine genetics as the blueprint of life. It's a complex and fascinating field that studies how traits are passed from parents to their offspring. These traits, which can range from eye color to height, are determined by segments of DNA known as genes. Now, DNA is coiled up tightly into structures we call chromosomes. Humans typically have 46 chromosomes, 23 from each parent. These chromosomes are paired into 22 autosomal pairs and one pair of sex chromosomes. The sex chromosomes are the ones that determine the gender of an individual. Females have 2 X chromosomes, while males have 1 X and 1 Y chromosome. When it comes to reproduction, each parent contributes 1 chromosome to each pair. For the sex chromosomes, a mother will always contribute an X chromosome. A father, on the other hand, can contribute either an X or a Y chromosome. This is where the possibility of choosing the gender becomes a reality. If the father contributes an X chromosome, the offspring will be female. If he contributes a Y chromosome, the offspring will be male. This is where Preimplantation Genetic Diagnosis, or PGD, comes in. PGD is a procedure used during in vitro fertilization. It allows scientists to examine the genetic makeup of an embryo before it is implanted in the womb. This means that it's possible to determine the gender of the embryo by examining its sex chromosomes. If parents have a preference for the gender of their child, PGD can make that possible. But it's important to remember that PGD isn't just about selecting gender. It's also a powerful tool for detecting genetic disorders. By examining the chromosomes and genes of an embryo, scientists can identify potential genetic diseases like Down syndrome or cystic fibrosis before the embryo is implanted. Now that we have a basic understanding of genetics, let's explore the techniques used in PGD. The first technique that we will talk about is polymerase chain reaction, or PCR. Now, don't let that big scientific name scare you off. Imagine PCR as a tiny meticulous photocopy machine, but instead of duplicating documents, it copies specific sections of DNA. When it comes to Preimplantation Genetic Diagnostics, or PGD, PCR becomes an essential tool. Here's why. In the world of genetics, we're often dealing with incredibly small amounts of DNA. To make a reliable diagnosis, we need to be able to study the DNA in detail, which can be tough when we have so little to start with. That's where PCR steps in. PCR uses a special enzyme called a polymerase to create multiple copies of the gene of interest. It's like hitting the copy button on our imaginary DNA photocopier. This process is known as amplification, and it allows us to have a closer look at the DNA we're interested in. This is especially crucial when determining the gender of a fetus. The gender or sex of a baby is determined by 2 chromosomes, X and Y. Females have 2 X chromosomes, while males have 1 X and 1 Y chromosome. By amplifying the DNA, PCR allows us to identify these chromosomes and thus determine the sex of the baby. But PCR doesn't just stop at identifying the gender. It can also help identify any potential genetic disorders. For instance, if a certain gene is present in an unusually high number, that could indicate a genetic condition. PCR allows us to spot these anomalies early, offering expecting parents crucial information about their baby's health. So, in a nutshell, PCR is a bit like a magnifying glass for our DNA. It allows us to take a closer look at the genetic material, helping us identify not just the gender of a baby, but also potential health concerns. PCR is a powerful technique, but it's not the only one used in PGD. Next, we will discuss fluorescent in situ hybridization. Scene script. FISH, or fluorescent in situ hybridization, is another technique employed in PGD. Imagine an artist working meticulously to craft a portrait. They use different colors to highlight different features, bringing the image to life. Now replace the artist with a scientist and the portrait with our genetic blueprint. This is essentially what happens in fluorescent in situ hybridization, or FISH for short. In FISH, fluorescent dyes are used to paint the chromosomes in a cell, each with a different color. This color coding allows scientists to count the number of chromosomes present in an isolated cell. Now, you might be wondering why we would want to count chromosomes. Well, it all has to do with health and disease. Every human cell should have 46 chromosomes, 23 from the mother and 23 from the father. But sometimes this count is off. An extra or missing chromosome can cause a range of genetic conditions. For example, Down syndrome is caused by an extra copy of chromosome 21. FISH is a powerful tool in identifying such chromosomal abnormalities. By painting the chromosomes and counting them, scientists can detect these abnormalities long before any symptoms appear. This is particularly useful in the context of preimplantation genetic diagnostics. Before an embryo is implanted during an IVF procedure, a single cell is removed and tested using techniques like FISH. This allows doctors and parents to ensure that the embryo is chromosomally healthy, reducing the risk of genetic diseases. Moreover, FISH can also help in identifying the gender of the embryo. Remember the 46 chromosomes we talked about? 2 of them determine the gender. Females have 2 X chromosomes, while males have 1 X and 1 Y chromosome. By identifying these gender chromosomes, FISH can be used to choose the gender of the baby in gender-specific genetic disorders. Together, PCR and FISH form the backbone of PGD, allowing parents to choose the gender of their baby. Now that we've covered the science behind PGD, you might be wondering about its implications. The use of PGD for gender selection is a topic of intense debate, often centering around ethical considerations. Some argue that it's a natural extension of parental rights, while others express concern about the potential for gender imbalance or misuse. Practically speaking, PGD is a complex procedure that requires considerable resources. It's not a decision to be taken lightly, but it does offer potential benefits. These include the ability to avoid certain genetic disorders and to plan for a balanced family. However, it's essential to remember that PGD is just one tool in the vast arsenal of reproductive technology. It's not a magic bullet, nor is it the right solution for everyone. So that's a wrap on PGD and how it's revolutionizing the way we think about parenthood. Remember, science is always evolving, and so is our understanding of it. If you found this video informative and interesting, don't forget to hit the like button and share it with your friends.